Biochemistry 401, Lecture 31. Today we're going to talk about protein degradation. We'll begin with dietary proteins and then work on to cellular proteins. We're then going to talk about amino acid catabolism. This includes the urea cycle. We're then going to finish with inborn errors of amino acid metabolism. The digestion of dietary proteins begins in the stomach. This occurs through the action of an enzyme called pepsin. Pepsin is originally synthesized as an inactive zymogen called pepsinogen. Pepsinogen includes a 44 amino acid portion that's stuffed into the enzyme active site. And so in order for this enzyme to be activated, 44 amino acids must be clipped away. This happens when the pepsinogen hits the acidic environment of the stomach. The low pH allows that protein to unfold just a little bit because there are amino acids that become protonated. And so the cell bridges that were there, the ionic bonds, are no longer there. And so the enzyme opens up just enough for these 44 amino acids to become vulnerable to proteolysis. And so pepsinogen autoproteolizes and the 44 amino acids are gone and the pepsinogen becomes activated to pepsin. Which of these amino acids has an R group that you might expect to go from a non-protonated to a protonated state following a shift from pH 7.3, what you would find inside a cell, to about pH 2. This is about the pH of the interior of the stomach. So which of these, lysine, glutamine, aspartate, alanine, or tyrosine? That's right, it's aspartate. There are actually aspartates in the active site that are responsible for the catalytic activity of this enzyme. And so when aspartate becomes protonated, it's no longer going to bind to a lysine. Therefore, the protein can open up just a little bit to become vulnerable to proteolysis. And so this 44 amino acid segment that is present in the active site is going to be cleaved away to activate this enzyme. Now the thing with pepsin is, it is a random cleaver. It just goes along and it chops up proteins randomly, non-specifically. And so there's not an amino acid sequence that it favors. It just goes after proteins and proteolyzes them. The thing is, once this pepsin reaches the duodenum is a different place. It's the first part of the small intestines. You have bile coming in. Now, bile doesn't just have products from the liver coming in, not just bile salts. It also has pancreatic juices, it's called. Oh. These contain proteases also that are secreted from the pancreas as zymogens. And then once they get to the duodenum, they are activated. Bile also contains bicarbonate. And the bicarbonate is going to raise the pH of those stomach contents that are coming from the stomach into this first part of the small intestine. And so this bile is going to increase the pH of the food as it travels into the duodenum. This means that pepsin is going to be deactivated. It does not do well in a high pH environment, and so it's going to become deactivated. This is really good for the proteases that are coming in from the pancreas. Now they're not going to be randomly cleaved by this pepsin. Awesome. Once pancreatic zymogens get to the small intestine, there is a protein on the outer surface of the enterocytes. So we have the small intestine. Okay, let's say this is the surface of my small intestine, and we're going to have a protein on the surface that is called enteropeptidase. This is a transmembrane protein that has a protease activity that extends into the lumen of the small intestine. So when the zymogens come in from the pancreas, you first start off with trypsinogen. Now trypsinogen is the zymogen. You know it's a zymogen because it has O-gen at the end. These zymogens are either going to have O-gen at the end or pro at the beginning. That's how you know. So here comes trypsinogen. It's going to encounter enteropeptidase. Enteropeptidase is going to cleave off the inhibitory portions of trypsinogen to make it into trypsin. Now trypsin is going to go along and activate all of those zymogens that come in from
the pancreas. So this is like chymotrypsinogen, this is proelastase, and you can see right here in the diagram that there is a cascade. Trypsinogen is going to be activated by enteropeptidase, it's going to become trypsin, and it's going to activate the proteases in this cascade. Now the thing is that these proteases are specific cleavers. They hydrolyze the peptide bond at specific amino acid sequences. Why? Because these particular sequences fit into the active sites of these enzymes. This has to do with the architecture of the active site itself in these proteases. Active site specificity. Let's take a look at that. For instance, trypsin has an aspartate in the active site, whereas elastase has a couple of valine in the active site. And it is the nature of the active site that determines its specificity. Each of these proteases, chymotrypsin, trypsin, and elastase, cleave the peptide bond after a specific amino acid that fits into the active site. Can you pair these proteases up with their target amino acid? Because trypsin has a negatively charged amino acid, aspartate, in its active site, it's going to be specific for positively charged amino acids like glycine and arginine. Chymotrypsin has a large active site without any bulky hydrophobic residues, without any charged residues in it. So it is going to cleave after large hydrophobic amino acids like phenylalanine or tyrosine or even tryptophan. Elastase likes to cleave after an alanine or a valine. Do you have to remember these specificities off the top of your head? No, but if I give you the active site, you'd have to be able to match that up with the type of amino acid that it would cleave after. And so once these proteins are cleaved by these proteases, both from pepsin and pancreatic proteases, they're taken into the enterocytes, the epithelial cells lining the gut, the small intestine. And then they're broken down even more by peptidases that are in the enterocytes. Finally, these are shuttled in the bloodstream as amino acids. What can we do with amino acids? Well, we can use them to make other proteins, right? We can also use them to make other metabolites that we need. But another thing we can do is we can break those down, take the amino group off, and then we have a carbon skeleton, right? And so we can use that carbon skeleton in anabolic reactions and for energy. So now we're going to turn to the breakdown of endogenous proteins. There are times that we have to break these down. Why? Well, one of the ways we control the activity of enzymes is by cleaving them, getting rid of them, and reducing their concentration in the cell. So we can regulate enzyme activity by getting rid of some of them. Another thing we have to worry about is proteins that are misfolded. Now, these can either be proteins that never made it when they were being translated. They just came off the ribosome a little wonky and they never folded right. Chaperone couldn't help them, and so they're going to be degraded. Same thing for proteins that have gotten damaged in the course of their lifetime and now they can't fold properly. They can also be degraded. If they can't be refolded by a chaperone, they get degraded. Another thing that's important to know is that proteins have characteristic half-lives, right? They have sequences at their ends that cause them to either be really stable proteins and hang around for a long time, or to be unstable proteins and have a lifespan of only a certain amount of time. And this is an intrinsic property of the protein itself, caused by its sequence. Another reason we would degrade proteins is so that we can use those proteins for fuel. We can break down proteins and get the amino acids and then catabolize those in order to gain energy as we feed those carbon skeletons into the TCA cycle. And as we said before, we can also use these amino acids for anabolic precursors. So let's look at how proteins are degraded in the cell. As you can imagine, it's something we have to be really careful of. We want to make sure that we degrade the right protein at the right time. Otherwise, we can get in big trouble, right? Okay, let's take a look at that. So we talked about ubiquitination before. This is the addition of ubiquitin proteins to a protein that's being degraded by the proteasome. Ubiquitination happens through the action of three enzymes. E1 activates the ubiquitin, E2 binds to the ubiquitin, and E3 transfers the ubiquitin to the protein. E2 and E3 are very specific for specific targets. They have an architecture that is complementary to a certain protein shape and causes its ubiquitination and its transfer to the proteasome. The thing is we don't just have one ubiquitin that's attached, but we'll have a whole string of them, at least 4 or more that are attached to a protein in order to target that for destruction. The proteasome has 3 main parts. It has one cap at the top, it has a barrel, and it has another cap at the bottom. The caps at the top and the bottom are called 19S, and the portion in the middle is called 20S. You put them all together, and you get the 26S proteasome. These are in sedimentation units. You don't add 19 and 19 and 20 and say, well, how did I get to 26? It's all sedimentation units, Svedberg units. The ubiquitinated protein comes to the proteasome, and there are regulatory proteins that are involved with the cap itself that help to bind that protein and take off the ubiquitin and feed the protein into the barrel of the proteasome. The barrel itself is where the protein is going to be cleaved through attacks on the peptide backbone. So clip, 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 and then the bottom opens and the fragments go out. These are then acted on by other proteases that cleave them into individual amino acids that can be broken down through a series of catabolisms that are specific for specific amino acids. There's a general theme in amino acid catabolism whereby we release an amino group from an amino acid and we transfer that to an alpha keto acid, generally alpha ketoglutarate. Then alpha ketoglutarate can carry that amino group safely in the bloodstream to the liver. Why? Because most tissues don't have the urea cycle, and the urea cycle is our main way of getting rid of ammonium. Ammonium is a nasty thing. If it builds up, it can cause real problems. It causes things like lethargy, nausea, hallucinations. Then it can actually lead to coma and death. Really scary, really nasty stuff. You can see high ammonia levels in people who have a compromised liver. If they're having a hard time with their liver for some reason, one of the problems that a person can have is a problem with disposing of amino groups through the urea cycle. High ammonia levels can result. So we need to have a really safe way of getting rid of those ammoniums, not just transporting them as free ammonium, but getting them on a specific carrier to carry them to the liver for disposal by the urea cycle. And so the process by which we transfer an amino group is called a transamination. That makes sense, right? We're going to transfer an amino group from an amino acid to an alpha keto acid. This can either be pyruvate, or it can be oxaloacetate, or it can be alpha ketoglutarate. Most of the time, it is alpha ketoglutarate. That's a major transporter of amino groups.
There are 2 important transaminases. The first one is aspartate transaminase, AST, also known as aspartate aminotransferase, AAT. In this reaction, an amino group from aspartate is transferred to alpha ketoglutarate to yield oxaloacetate and glutamate. Now we saw this enzyme when we looked at the malate aspartate shuttle. Another important aminotransferase is the alanine transaminase, abbreviated ALT. This is also known as alanine aminotransferase, abbreviated ALAT. In this reaction, alanine transfers its amino group to alpha ketoglutarate to yield pyruvate plus glutamate, and this is an important part of the alanine cycle. This is the major way in which muscles release amino groups safely for transport to the liver. The alanine aminotransferase is part of the glucose-alanine cycle, which is also known as the alanine cycle. And this is an important cycle that the muscles use when they're required to exercise, even when there isn't very much glucose in the blood. Under fasting conditions like this, the muscles will break down glycogen and branched-chain amino acids in order to yield energy. Glycogen will be broken down first to glucose 1-phosphate, then glucose 6-phosphate, finally to pyruvate. This is an alpha keto acid that can accept an amino group from the branched-chain amino acids. Now, we want to give this amino group from the branched-chain amino acids to pyruvate in order to transport this safely as alanine. Now, why is this? It's because muscle cells do not have the urea cycle. They cannot eliminate nitrogen safely as urea. And so these nitrogen-containing amino groups must be gotten rid of another way. So they're transferred to pyruvate, which makes alanine, and the alanine goes through the blood to the liver. Once in the liver, alanine is transaminated by passing its amino group to alpha-ketoglutarate to yield glutamate. The amino group can then be hydrolyzed off and used in the urea cycle to be eliminated safely. Now, once alanine gives off its amino group, it's turned into pyruvate again, and then pyruvate can be used as a substrate for gluconeogenesis, and this glucose can enter the bloodstream to feed the body as a whole and to feed the muscles as well. In this way, the cycle is completed.
So the thing is, the transaminase reaction happens in 2 steps. First, an amino group comes into the enzyme, and the enzyme takes off its amino group and holds on to it and goes off as an alpha keto acid. Then an alpha keto acid comes in, picks up that amino group, and goes off as the other amino acid. So for instance, let's say aspartate is coming in. It's going to come in, here it is, aspartate. It's going to drop off its amino group, and it's going to leave as oxaloacetate. And here it goes. Here comes alpha ketoglutarate. It's going to come in, it's going to pick up that amino group, leaves as glutamate. And that's how aspartate aminotransferase works. What holds on to that amino group? Oh, that is very cool. It's something that's called pyridoxal phosphate, and it makes a shift base to the amino group and holds on to it. Once you break down these amino acids, you get a specific group of metabolites that are formed. There are 7 of them, and they're shown here.
Pyruvate, acetyl-CoA, acetoacetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate.
You do need to know these because they're really important intermediates, and we've seen so many of these before. Many of them are intermediates in the TCA cycle. These will feed into the TCA cycle at certain points. Then the liver has its favorite food. It likes to break down carbon skeletons in the TCA cycle. So it's all good. We can also use these carbon skeletons for anabolic precursors to either be available for ketone body generation or to be able to be made into glucose. So some are ketogenic and some are glucogenic, and some are both. The thing is with ketogenic amino acids is the fragments that they break down into only have 2 carbons. And so they feed into the TCA cycle at the level of acetyl-CoA. That's why they can't be used to make glucose. Not enough carbons. They come in, 2 carbons go out, no net carbon gain. That's why they're ketogenic. The ones that are glucogenic break down into components that are more than 2 carbons in length. So pyruvate, for instance, oxaloacetate, alpha-ketoglutarate, things like that. There are some amino acids that when they break down, they have both a 2-carbon component and a component that's more than 2 carbons. And so they can feed into the TCA cycle in portions that are both at the level of the acetyl group and then levels that can actually make glucose. And so those are both ketogenic and glucogenic. Here's a list of them. And they're really cool mnemonics to remember which are ketogenic and which are glucogenic and which are both. Here we go. The ketogenic amino acids are the 2 L's, lysine and leucine. Pretty easy. The ones that are both, just remember, Phil is tripping on tires. Phil, phenylalanine, is isoleucine, tripping, tryptophan, tires, tyrosine. Phil is tripping on tires. Those are the ones that are both ketogenic and glucogenic. The ones that are just glucogenic only are everything else that I didn't mention. So not the 2 L's, not phenylalanine, not isoleucine, not tryptophan, and not tyrosine. There you go.
So the last step before the urea cycle is the oxidative deamination to remove the amino group from glutamate to yield alpha-ketoglutarate. This is done through the action of glutamate dehydrogenase. The first thing that happens is an oxidation that requires NAD+ or NADP+. This forms a shift base intermediate. The next thing we're going to do is we're going to hydrolyze that amino group right off through the addition of water. This is a hydrolytic deamination, and this yields alpha-ketoglutarate and ammonium. Glutamate dehydrogenase is a mitochondrial enzyme. It's best to keep ammonium safe and locked away. Now, this reaction, as I said, can use either NAD+ or NADP+ as an electron carrier. In animal cells, under normal conditions, the reaction catalyzed by glutamate dehydrogenase is close to equilibrium. And although it could easily go forward or backward depending on the concentrations of products and reactants, it's usually pulled forward in animals by the loss of ammonium to the urea cycle. Here we see the structure of urea. The first amino group comes from ammonium, the second amino group comes from aspartate, and the carbon comes from bicarbonate. The first reactions occur in the mitochondrial matrix, and the first major intermediate is carbamoyl phosphate. Carbamoyl phosphate is synthesized by carbamoyl phosphate synthetase, and this is a major point of regulation in the urea cycle. The first thing that happens is the activation of bicarbonate with ATP to form carboxyphosphate. Next is an addition of ammonium to form carbamic acid, with the release of inorganic phosphate. Finally is the activation of carbamic acid with ATP to form carbamoyl phosphate. Now this is a high-energy intermediate because it has an anhydride bond. Therefore, this particular molecule has a high transfer potential. This enzyme is called carbamoyl phosphate synthetase, and is called synthetase because it requires ATP. Now this enzyme works through what's called substrate channeling. These intermediates are passed from one position to another within the enzyme. First, bicarbonate is phosphorylated, and it travels to the next place for the addition of ammonium. Then finally, there's an addition of ATP. Substrate channeling prevents the loss of intermediates so that ATPs are not squandered. The next step is to combine carbamoyl phosphate and ornithine to yield citrulline. Again, this happens in the mitochondrial matrix. The enzyme that catalyzes this reaction is called ornithine transcarbamylase, and again, this name makes sense. We're going to transfer carbamoyl from carbamoyl phosphate to ornithine, and we're going to release inorganic phosphate. Both ornithine and citrulline are in fact amino acids. They have an alpha amino group and an alpha carboxy group, and an R group. These, however, are not used in proteins. Now, with the formation of citrulline, we have a product that's going to exit the mitochondrial matrix and enter the cytoplasm. Once in the cytoplasm, it's going to combine with aspartate to yield argininosuccinate. This enzyme requires ATP and is driven forward through the formation of pyrophosphate and subsequent hydrolysis to inorganic phosphate. The name of this enzyme is argininosuccinate synthetase. Again, it's called synthetase because it requires ATP. The product of this reaction is called argininosuccinate. And if you look at it closely, it's arginine with just a little bit extra. This little bit extra is actually given off in the next step that is catalyzed by argininosuccinase to release arginine and fumarate. Fumarate is a link between the urea cycle and the TCA cycle. We're now going to cleave off the guanidinium group from arginine to form ornithine and free urea. This is catalyzed by the enzyme arginase. Ornithine, of course, can be transported to the mitochondrial matrix to complete the cycle, and urea is given off in the urine. And so, the urea and the TCA cycle have a direct intermediate in common, and this is fumarate. Through hydration, this can be used to make malate, and malate can be oxidized to make oxaloacetate. And then we can transfer an amino group to oxaloacetate to yield aspartate again. And then aspartate can enter the urea cycle again. And in this way, we can keep the cycle going with a good supply of aspartate. And now we're going to switch and talk about the catabolism of specific amino acids that are associated with inborn errors of metabolism. These are errors that give rise to some pretty awful diseases. These include phenylketonuria, alkaptonuria, and maple syrup urine disease. First, we're going to talk about the catabolism of aromatic amino acids, phenylalanine and tyrosine. and then we're going to talk about the catabolism of the branched-chain amino acids, valine, leucine, and isoleucine. The catabolism of phenylalanine feeds directly into the catabolism of tyrosine. The first thing that happens is a hydroxylation to make tyrosine, and this is catalyzed by phenylalanine hydroxylase. This is an important enzyme to remember. The next thing that's going to happen is a deamination to form hydrophenylpyruvate. The next thing that happens is the formation of homogentisate, and this is an important intermediate. Now the next step requires homogentisate dioxygenase to make 4-mallyl acetoacetate. Homogentisate dioxygenase is another important enzyme to remember. The product of this reaction is 4-mallyl acetoacetate. This eventually is going to be broken down into two products: acetoacetate that can enter the TCA cycle at the level of acetyl-CoA, and fumarate. In the catabolism of phenylalanine, the conversion to tyrosine by phenylalanine hydroxylase requires an electron carrier, tetrahydrobiopterin. But unlike FAD and NAD+, tetrahydrobiopterin is not vitamin-derived. This is because we can make this ourselves. Now, phenylketonuria is an autosomal recessive disease that results from a mutation in phenylalanine hydroxylase or insufficient concentrations of tetrahydrobiopterin. Mutations in phenylalanine hydroxylase occur approximately 1 for every 15,000 live births in the U.S. Deficiencies in the electron carrier, tetrahydrobiopterin, however, are rare. If phenylalanine cannot be turned into tyrosine, it's turned into phenylpyruvate instead. One of the things about metabolism is if you can't do things the right way, there are plenty of wrong ways to do things, and this is evidence of one of them. Because the outcomes for phenylketonuria are severe neurologic and cognitive impairment if untreated within the first year. We have been screening infants by blood test for this disorder since the 1960s. The treatment for phenylketonuria is a restricted diet that's low in phenylalanine and high in tyrosine, and you can think about why that would be. Alkaptonuria is another problem associated with the breakdown of phenylalanine. In this case, it's an absence or a deficiency of the enzyme homogentisate oxygenase. And because homogentisate cannot be turned into 4-mallyl acetoacetate, it's excreted in the urine. Once the urine hits air, homogentisate can become oxidized, and this makes the urine black. This is also called black urine disease, but it's a relatively benign disease because homogentisate can be gotten rid of through the urine. And the last thing we're going to talk about is the catabolism of branched-chain amino acids. This is the breakdown of valine, leucine, and isoleucine. We know that this catabolism occurs in the muscles in order to generate energy. The first two steps in the catabolism of all of these amino acids are the same. The first thing that's done is a transamination, and this is catalyzed by the branched-chain amino acid transaminase, and alpha-ketoglutarate is the amino group acceptor. This yields glutamate and 3 different alpha-keto acids. The next thing that happens is an oxidative decarboxylation of the alpha-keto acids. This happens through the action of branched-chain alpha-keto acid dehydrogenase complex. It yields 3 different CoA derivatives. This complex is similar to the pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex. The next disease is called maple syrup urine disease because the urine of these individuals smells like maple syrup. But this is actually a really terrible disease caused by a defect in the branched-chain alpha-keto acid dehydrogenase complex. And so the oxidative decarboxylation of the alpha-keto acids from the branched-chain amino acids does not occur. And there's a buildup of these alpha-keto acids and also the branched-chain amino acids. This is a really bad thing that leads to severe mental deficiency and a failure to thrive. This is also identified early on through newborn screening in the hospital, and these individuals also have a restrictive diet.
This is the end of Lecture 31. I hope you enjoyed it, I hope you learned a lot, and I hope you have a great day.